All right, so now let's go to the different classes of immunoglobulins, because remember we have IgM, D, G, E, and A. Yeah, let's go through the way in which we make these different versions, because all of these can share the same V, D, and J regions. It's just you're swapping out the constant region at the end, which is going to be roughly equivalent to the FC fragment, or it's going to contain the FC fragment. Okay, so there are different immunoglobulins, and yeah, they have slightly different structures. So M, D, G, E, and A are shown here. All the little hexagons are sites where we have sugar molecules attached. And so you can see there's different patterns of sugar molecule attachment, and there are some proteins that can recognize and differentiate different types of immunoglobulin based on where the sugars are and how many there are and what kind they are. Yeah, and so for humans, well, all right, we can start with mice. So we're going to have all of our V, D, and J up here, and then we have a set. So it's like a bank of constant regions. And the arrangement's a little different in mouse and human. Actually, no, let's focus on the human. So they're going to be in roughly the order in which we make them. So constant region mu is for IgM, and then constant region delta is for IgD. And we're actually going to make those at the same time from the same piece of messenger RNA, from the same transcript. We will just splice it differently to leave out the mu or to join it up to the mu and never use the delta. That's the idea. But we can undergo further changes, and we can actually express it with an IgG or E or A constant region. And if you notice, there are actually multiple versions of IgG and IgA encoded in the human genome. And so we've got IgG1, IgG2, IgG3, and IgG4. So that's made by the gamma 1, 2, 3, and 4. See the little thing I'm pointing at? And yeah, depending on which one of those we get, they will make slightly different versions of antibodies. They can bind to slightly different proteins. They bind, better or less well to different proteins. There is a pseudogene here for a second copy of IgE. And the little Greek letter psi in front, that's what this thing is. It looks like a little trident. Tells you this is a pseudogene. And so you can kind of see what's happened here, right? We started out with a real basic set like they have in the mouse with just two IgG genes, and then an IgE and an IgA, and we duplicated that whole thing. So we've got one copy here, and we've got another copy here. It's just that over time, mutations have crept in, and we've lost our second IgE, which is actually just fine because IgE is one of these antibodies that doesn't have a lot to do anymore. It is going to directly bind to things like mast cells and eosinophils, and it's going to set off horrible allergic reactions that could have potentially helped in the past when we led shorter and more parasite-ridden lives. But now, thankfully, in the modern world, we don't necessarily have to deal with that nonsense, and that's a very good thing, I would say. Okay, so how many different classes of, or let's compare the different classes of immunoglobulins, let's say. And each of these is better or worse at different things. So, yeah, fantastic. So look who's good at binding at high affinity to mast cells and basophils in addition. That's going to be IgE. And mast cells and basophils are both going to have histamine and other granules inside there, and they're going to release those and cause very strong immune reactions like you would get in an allergy, just unnecessarily. IgM is going to be the best at activating the classical complement pathway, and this is good. IgM is going to be the first antibody, that we actually make, and so it's good if it is a strong activator of complement, because if we're just starting to deal with a bacterial infection, this lets us get off the ground as fast as possible and just beat up the bacterium or virus before it can really do anything. Now, IgG 1 and 3 tend to be pretty good at complement activation as well. They're going to be the ones that are most common in the bloodstream. And they're also going to be the ones that get transferred across the placenta and will end up inside the baby if you happen to be growing a little baby inside you right now. Let's see, IgG2 and IgG4 are not really great at either of those things. They can stick to a pathogen, but they're not going to do a whole lot else, and they're not going to be particularly abundantly transferred across the placenta. Now, if you look at macrophage and other phagocyte like neutrophil receptors, they're okay at binding any of these things. So that's what I was saying, like an antibody is like one of those cocktail toothpicks that you can use to pick up anything off the hors d'oeuvre tray. So these are going to let any old white blood cell basically pick up whatever the antibody is bound to and eat it, consume it, and digest it. So, yeah. Okay. All right. Next unknown term is ZFP318. So this is going to be a protein involved in splicing. This is part of a thing called the spliceosome, which is a complex of proteins and little pieces of RNA that get together. And together these are going to mark where you should do the splicing and actually do the splicing themselves. And by splicing, I mean taking out the parts of an RNA that you're not going to use. So throwing away the introns and keeping the exons. Yeah, and so these are going to lead to expression of IgD on the surface of a cell. It's just through differential splicing. We're going to take a message and we'll splice it different ways and make different products from it. All right, so how's this work? Well, we can make a, on the piece of DNA we're going to recombine the V, D, and J so they're all joined together. So they're going to be this little red end, which is going to make the heavy chain variable region shown in this particular diagram. To express that, we're going to make a messenger RNA that has the VDJ region and the constant region for IgM and the constant region for IgD together on there. And so we can make the short RNA that's only able to make IgM, or we can make the long RNA that has the M and D genes, constant regions on there. And we can actually just loop out and get rid of this IgM during splicing. And so that's what this crazy protein, ZFP, zinc finger protein 318, is actually doing. So it's the one that's involved in this particular bit of splicing. And so without that, you can't make IgD and mount it on the surface of your immune cells. But yeah, that comes along with a fairly severe immunodeficiency. So yeah, this is a good thing that we have this particular protein, this zinc finger protein. All right, next one. So CSTF proteins are cleavage stimulation factor proteins. And they're going to be involved in splicing as well. They're going to be modifying the end of a messenger RNA, or a pre-messenger RNA rather, so that you can put a poly-A tail on it. Because a messenger RNA is going to have two things on it. It's going to have a cap at the front end, or 5' end, and it's going to have a polyadenosine, just a lot of A nucleotide tail at the 3' end, which is like the tail of it. And yeah, this is going to be necessary if we're going to translate a protein from a piece of RNA. And since that's the only point of messenger RNA, yeah, this is a thing that has to happen in messenger RNA production. So you need cleavage stimulation factor number 64. That's the one that's going to add a poly-A tail that will lead to the secreted form of IgM. And the secreted form is the neat one. That's the one that chains together. There's going to be 5 of them hanging out together in most cases. Last little gene we need to talk about is an elongation factor. So this is elongation factor L2 or ELL2. And this is going to promote poly-A addition, leading to the secreted form of IgM. So yeah, together CSTF64 and ELL2 are going to do the job. So how do they do this? Wow, just like this. So we've got our leader region, we've got our VDJ region, we've got our mu chain here, which is going to come and 4 different exons, and we're going to get rid of those little introns in between by splicing. And then we're going to splice out these little bits at the end and join the yellow bits on. They're going to make the tail. So the M1 and M2 regions are actually going to add a transmembrane anchor to a protein. And so this is how we make transmembrane IgM. We just splice those so that they get added on here. And if we want to make secreted IgM, we basically just stop splicing at the end of this, and we get rid of those yellow regions so that we do not make the little membrane bit that hangs off. And so, yeah, you end up with a antibody that is not bound to the membrane. The antibody bound to the membrane serves an important purpose. So this lets the rest of the immune system know what kind of antibody this cell has decided to make. And so then the rest of the immune system can either reward that if it's making a useful, good antibody, or they can not reward it and cause the cell to kill itself if it's not making anything that is useful at the moment. And so that's how we can just keep cycling through, keep trying out new combinations. And then when we get to a time where we need one, we start selecting and make different versions of them. Okay, a J chain is like a little joining chain. So this is different from the J region of VDJC recombination. The J chain or joining chain is the one that's going to stick multiple copies of an antibody together. So it's going to attach 5 IgMs to each other or 2 IgAs to each other. And it's going to do this with some disulfide bonds. And so here you can see the little J chain, and the disulfide bonds are shown here in black. And so together these are going to hold the molecule together. And we're going to end up joining up 5 different IgMs or 2 different IgAs when we do this. And it basically happens because the joining chain is going to come in here, and ordinarily these two, I think, would be bound to each other, disulfide bonded to each other. But now they can't because they're bound to the join chain, and so they're just going to keep bonding to each other. And the structure of the molecule itself lends it to this geometry. It forces it to be 5 copies, and the structure of the IgA is a little different, and it's going to force it to be 2 copies. All right, so that's the first part of this one. Now let's do 5, 19, 20, and 21, which is just a little bit of interesting extra stuff. So we are not going to cover 517 or 518 on the test, but it's kind of neat from the perspective of evolution if you're interested in where these things come from. So we'll pause here, and I'll make this a separate part where we go through these things. Because we're talking about, just how did we get to this situation where humans have this awesome array of T-cell receptors and immunoglobulin genes.